In this video, we're going to discuss the back half of the Introduction to Biology notes, where we start to talk about the process of science. This topic is also covered in lab at some point, so there will be a lab that helps you understand how a science experiment gets run and sort of what is the process that's involved in that sort of thing. But to start us out in the notes, we have the question, what are some of the methods of science that are mentioned in the book? And this is them, so get this stuff that's up here on the screen in your notes. As always, if I talk too fast for you, remember you can pause the video to make sure you still get what you need up there. Anytime you're conducting any kind of science experiment, one of the things that you have to be really good at is observing things that you might not even have thought needed to be observed. Sometimes little things that actually matter can be ignored, and then scientists don't realize that until later on. For example, there were scientists who were trying to recreate Pasteur's experiments where he disproved spontaneous generation, but because they did it in a different setting, they kept getting different results, and it was because they didn't observe the fact that the area that they were working in was dirty, and so it kept contaminating their results. One of the other ones that tends to be something that students struggle with these days is accurate record keeping. If you're going to do science, you have to be very organized and you have to keep accurate records of everything that you're doing. If you have something that's improperly labeled, it's going to tend to mess with the results because you thought that what you were working with was something other than what it actually was. And so be organized, be accurate, and then take detailed records or collect data as you do your experiment. Next, logical and mathematical reasoning. If you were to read any sort of journal article, you would find that there's some element of statistics that gets run in that article. Usually, at the bare minimum, we're going to be calculating something called standard deviation. Usually we're also going to run probability tests like ANOVA or student t-tests, and that helps us to verify that our results are actually meaningful. Sometimes just looking at an average or a percentage might make you think your data means something, but unless statistics get run, you don't know really if you're within the standard deviation or not. Running the experiment means that you're going to be testing variable groups and control groups, and submitting conclusions to the scrutiny of others means you write your results up like a lab report, and then you submit that, and then when you submit it to a journal, they send it out to reviewers. The reviewers read your paper, and then they make you wish you were dead, essentially, because they tell you everything that you did is wrong, and you need to re-edit this and add more citations here and do all of these things. But they're doing that to make sure that the whole process would actually stand up against scrutiny. And what I mean by that is, they thought of things that maybe you didn't think about as you ran your experiment, and if you had thought about those things, it might have changed your results. And so it's always good to have somebody else look over your results just to make sure that you drew a valid conclusion from those. What are some of the things that methods of science cannot be applied to is what you have next. You can't apply them to political or moral views. Those things change over time. If you go back to the 1700s, the founders of this country didn't believe that women should own property or have the right to vote. If I remember right, they considered blacks to be 7/8 of a person back then. It was some fraction, and I'm sorry if I'm wrong about what that fraction was. As the nation has matured, our morals and our ethics has changed so that now women have the right to vote. Blacks have the right to vote, even though there are some places that are trying to disenfranchise them in today's modern era. But oh, my son just texted me it was 3/5, not 7/8. See, I didn't do very well in my history classes, so there you go. But those sorts of things change over time, and so you can't test those because what is a viable result today wouldn't be the same as what it was in the past, or it won't be the same as what it was in the future. Same for aesthetics and beauty. If you look at art, what they considered to be an attractive person in ancient Egypt isn't the same as what Ireland thought was attractive 100 years ago, and it isn't the same as what we thought as attractive now. Like, fashion stuff changes, and so does aesthetics. Nowadays, you have women who are doing things like getting butt implants and injecting silicone into their lips, and that would have been just insane 200 years ago for people to do, and so beauty changes over time as well. Religion backs up against two things. First, there's the moral aspect to it, and again, morality changes over time. And second, when you start talking about religion, whether you're talking about somebody that is polytheistic and believes in multiple gods or monotheistic and believes in one God, you can't prove the existence of God and you can't disprove the existence of God. And if you can't run an experiment on something to prove it, or at least provide evidence to support it, that means you can't run an experiment to do that, and you can't support or reject that idea. And so religion is something that can't be tested from a scientific standpoint either. Next step, define the scientific method. So your definition is up there. Once again, make sure you get that. Essentially, the scientific method is a way for you to test some question that you have. It's something that honestly people do all the time. They just don't think about it as a scientific experiment. Just to give you an example, let's say you were going to go out to your car and your car didn't start. You'd have a question about why doesn't your car start, and then you'd start thinking about possible solutions like, did I leave a light on in the cabin of my car so my battery's dead? Well, let's test the battery, or let's try to jumpstart the car and see if that works. That's running an experiment to try to see if the battery was actually a problem. And if you found that the battery wasn't the problem, well, then you come up with another idea, and maybe it's the alternator, maybe you ran out of gas, who knows? But you keep going until you find out what the actual problem is. It's just that when it's the battery that's dead in your car, you don't write up a report and then send it to a journal to see if somebody would peer review that for you. But scientific method is something that everybody does, they just don't usually call it the scientific method. What is a hypothesis? So once you start that science experiment to try to answer a question that you have, the hypothesis is what you think the answer to the question was. So once again, if your question was, why didn't my car start? Your hypothesis might be, well, maybe the battery is dead. That's a valid hypothesis that you can then test by either getting a voltmeter and testing the battery, or by trying to jumpstart the car and see if that's going to solve the problem. Based on what you read in the book, should it be a question or a statement? It's a statement. It's the answer to the question that you're trying to answer. And so it's the idea of what you think that answer will be. You run the experiment to then find out whether or not you're right. So what's going on in this picture is these women are digging in this field looking for something that is called yartsigumbu. Here's a picture of what it looks like. So this little brown stalk is what they're looking for in that picture. In some Asian cultures, it is believed that this stuff can do everything from cure baldness to curing cancer. And so they're looking for it because if they can find it, they can get a fair amount of money and then sell it to, I don't know if they call them apothecaries over there, but sell them to pharmaceutical sort of places. Here's the problem with yartsigumbu. It has very little scientific support behind it at this point. So if you did want to find out whether or not this substance could prevent cancer or cure cancer, you would need to get together a bunch of people who had cancer, give some of them yartsigumbu, give some of them a control, a placebo, and then try to see whether or not those two groups have any difference in them after the experiment gets run. If the group that ends up getting yartsigumbu ends up not having cancer at the end of it, but the group that got the placebo does still have cancer, that would be really good evidence that yartsigumbu actually works. But you've got to run the experiment to figure out if it works. And unfortunately, like I said, there's no science behind this particular thing. So hypothesis-based testing, that's how all drug trials work. Their hypothesis is this drug is going to work to treat XYZ diseases, and then they test that with control groups. What is a scientific theory? It is a thoroughly tested and confirmed explanation for a set of observations. The problem with the word theory at this point is that in the common vernacular, when somebody says, well, I have a theory that my battery's dead, that's not a theory, that's a hypothesis. A theory is something that has lots of evidence from hundreds of different experiments to help support it. So gravity is a theory. We know that if you drop something on this planet, it's going to accelerate at, 9.2 meters per second squared. We know that if you go out into space, there's microgravity up there. There's still gravity, but there's very little of it. But we also know that black holes do weird things to gravity. And so it is, and I'm doing quotation marks here, it's just a theory. We understand gravity. It has lots of evidence to support it, but it's just a theory. And I want to say that because I hear all the time as a biology teacher, well, evolution's just a theory, it shouldn't be taught. You need to remember that evolution has a lot of evidence behind it, from fossil evidence to biochemical evidence to biogeographical evidence. There's lots of evidence to support the idea of evolution. So it's not just a theory, it's a theory with lots of evidence to support it. A hypothesis is just a hypothesis. It's the possible answer to a question. So don't think of theory like it's a hypothesis. They are not the same thing. That really takes us to the next thing, and I was kind of talking about it, so I'll come back to this slide. How are theories and hypotheses different? A hypothesis is a day-to-day tentative answer for a question. It's something that, again, anytime you're running any kind of experiment, whether it's formal or informal, it's your possible answer to the question that you're actually testing. A theory, on the other hand, that has the support of hundreds of experiments behind it. And so a theory is more generally accepted than a hypothesis is. Looking at it another way, a hypothesis is an answer to one experiment. A theory is an answer to hundreds of experiments. And so again, just more broad-based support. Okay, after that, what is inductive reasoning? Once again, this is something that everybody does all the time, and they just don't know that it's called inductive reasoning. So when you're a kid and you start getting introduced to animals, usually with little books that are like, I don't know, A is for aardvark and B is for beaver, you start to learn certain things about animals. Like, most of the animals that you think of are probably mammals. They have fur, they have mammary glands. Every mammal you've ever seen more than likely has hair or fur, and so you associate animal with mammal with hair or fur. If you were to then try to draw a conclusion based on, well, hey, every mammal I've ever seen has hair or fur, that's the process of inductive reasoning. It's looking at all of these little pieces of evidence that you've collected and then making a generalization about all of that evidence. Well, every mammal I've ever seen has fur. That's how that is going to work. So that's how inductive reasoning works. What's the difference between qualitative and quantitative data? Qualitative means quality. So you're getting information like it's hot, it's cold, it's wet, it's dry. Words get used to describe qualitative data. Quantitative data, on the other hand, means quantity. It means numerical. You have a way to measure it. So if I were to ask you how this dog is feeling right now, you would qualitatively tell me, well, that dog's probably hot, and so it's laying in the ice to try to cool down. But we can be more specific than that, and in biology we want to try to be as specific as possible. So we could also get quantitative data by taking the temperature of this dog, and then we know exactly how that dog is feeling at this point. So quantitative data is generally a little bit more reliable because you have a number for it that got measured by something, and that just makes it more reliable. Now, I'm not saying qualitative data doesn't have value, it's just a little bit less reliable because you're usually judging that based on a person's experience. Like, how are you feeling right now on a scale of 1 to 10? That's more qualitative data. Okay, so we talked about inductive reasoning. That was forming generalizations based on your past experiences. Now we're going to talk about deductive reasoning. What deductive reasoning does is it takes the next step from inductive. So where in inductive reasoning you were making a generalization based on the past, deductive is, based on that stuff, make a prediction about the future. So deductive reasoning allows that prediction. So for example, let's say you've never seen this animal before. I chose it because not a lot of Americans have ever seen this animal. If you were to try to guess what kind of an animal this was, what would you choose? And I usually get one of two answers for this, and usually one of those answers is because somebody actually knows what it was. But if you had never seen it before, most of my students say it's a lizard, because it's got 4 legs, it's got scales, and so those two things put together based on their past experiences means this is more than likely going to be a reptile. However, if you were to flip this little girl over, she's got fur on her belly and mammary glands and nipples to feed that baby that is hanging out on her tail right now, so in actuality she is a mammal. So here's the reason I gave you this story. Deductive reasoning allows you to make a prediction. Sometimes those predictions are wrong. It doesn't mean you're dumb or stupid or anything. It just means that based on your past experiences, you had been led to believe that this might be the right answer, when in actuality, it's something else. Science has to change its mind all the time. One of the things that has been very frustrating for me as a scientist is hearing people say, I'm not going to wear a mask, the CDC has changed its mind too many times, they don't know what they're talking about, they're dumb. CDC changes its mind based on new evidence that comes out. They're not just at a whim deciding, oh, well, let's wear a mask now because I think they look sexy when it's 100 degrees outside. They're saying that because they looked at data from multiple countries and they found that countries that initiated mask laws had reduced the number of cases after the mask law went into effect. So that new evidence helps them change their mind. It's okay to change your mind. I encourage you to change your mind, because then at least you're looking at that evidence based on that. And so it's okay to be wrong as long as you start to change your mind eventually based on those errors. Okay. Next up, you just have a whole slew of questions to answer about the scientific method. Let's see. Know the process. Study figure 4. I think I'm going to show you figure 4 later on, by the way. Which comes first, the observation or the question? It's the observation. Like, you notice something, like, hey, my car didn't start. And then you ask the question, well, why didn't my car start? And then you can start running from that. But you got to observe something first. So this GIF, as it runs through a few times, it's showing you temperature data from the I think it was late 70s up until, 2016. What I want you to know is that the circle is getting wider over time, and that indicates the temperature has been getting warmer over time. Scientists have noticed that for decades. The temperature, on average, in the planet, within the atmosphere, and in the ocean, has been getting warmer over time. That observation led to the question, is the planet actually warming? Do we have a climate change that's coming? And then from there, we start to do experiments. And those experiments can be, let's measure the temperature in a lot of different places. Let's do it with satellites. Let's do it with temperature gauges in the ocean, even at the very bottom of the ocean. Let's do it in cities. Let's do it in the country. And then let's combine all that temperature data and see, is it actually getting warmer? Yes, it is. Let's also use, what about flowers and trees and things that are seasonal? Are they changing the time at which they start to reproduce? Like, are flowers flowering earlier or later? And yes, plants are flowering at different times. Since bees and butterflies don't use temperature to decide when to migrate, bees and butterflies aren't migrating they're migrating at the same time they used to, but flowers are opening earlier, which means a lot of them are starving to death. So the climate is changing, and it's affecting plants, it's affecting animals, it's affecting migratory patterns. Once again, we have lots of evidence to support the idea of climate change. It's not just one hypothesis anymore. We have dozens of hypotheses that have been confirmed, and the climate is changing. We have observed it. Is the hypothesis an if-then statement? A lot of high schools teach students that it is, but it's actually not. The hypothesis is broader than that. For example, going back to the car thing, you could say your hypothesis would have been, I think the battery's the problem. And then you have a prediction based on that hypothesis. If the battery's the problem, then jumping the car will fix the problem. And then if it does, you supported your hypothesis. So I really want you to see the difference between those. The battery being the problem is a hypothesis. It's a statement of what you think is the problem, and then the prediction takes it that step further to say how you can actually test that, and then what that test result would tell you. So that if then again just takes it that little bitty step further. Is the hypothesis the same as the prediction? No, it's broader, and you come up with the hypothesis first, then you make the prediction and you test that prediction. What does testable and falsifiable mean in reference to the hypothesis? Again, your definitions are up here, and those are pretty self-explanatory, so just make sure you get those definitions that are in here. These two things, incidentally, are why you can't test morals, ethics, beauty, religion. It's because you can't falsify or disprove God, so you can't disprove religion. And testable, you can't design experiments to test which of these is the most beautiful thing, and so some of those things, they're missing one or both of these, and so that's why you couldn't use science to prove them. Should you ever use the word prove in a paper? No. If you're writing an actual lab report, if you say prove, that means you're a cocky little person who believes nobody will ever show that you're wrong. And guess what? Somebody can always end up proving you wrong later on. So instead, if you actually conduct an experiment and it supports your hypothesis, you say exactly that. My data supported my hypothesis. But you don't say prove, because maybe you had an error in your methods. Maybe there was something you weren't thinking about. And so you just say support. That takes that cockiness out of it, so that it's okay if you end up being proven wrong later on. What are variables? These are the things that you're actually testing, and there's two different kinds of variables. There's independent variables and dependent variables. The independent variable is the thing that you have control of. Let's see, at this point I'm going to switch ideas and we're going to go back to that whole we're testing the Yartzagunbu to find out whether or not it can cure cancer. The independent variable would be, did the person get Yartzagunbu or did they get the placebo? That's the thing that you have control of, and it's the thing that you're testing. You're testing to find out whether or not Yartzagunbu can cure cancer. The dependent variable is the thing that you measure. In this case, it would be, did the size of the tumor shrink? Is the person in remission? It's those things that would tell you whether or not the Yartzagunbu worked. Saying that another way, the dependent variable depends on the independent variable. So the curing depends on the Yartzagunbu. That's why the curing of the symptoms is the dependent variable, and then the Yartzagunbu is the thing you're testing that makes that the independent variable. What is a control? So there's different kinds of controls. There's a control group. In a drug trial, that would be the group that gets the placebo. There's also controlled variables. Like, let's say that you were testing fertilizer treatments in plants, which I'm going to talk about an example like that here in just a second. You have to still make sure that all the plants are getting the same amount of sunlight. They're being held at the same temperature. They're getting the same amount of water. Those are controlled variables that would be kept constant between all the groups. So that's not a control group. It's a controlled variable. Okay, so here's an example of a controlled experiment that has all the features that an experiment is supposed to have. It has to do with crop rotation. So there are certain plants that can form symbiotic relationships or mutualistic relationships with bacteria. The bacteria can take nitrogen out of the atmosphere and turn it into essentially plant fertilizer. So if the plant can form that relationship, the plant gets fertilized by the bacteria. Not all plants can do that. Only legumes, which are beans, peas, stuff like that, are able to have that relationship with this kind of bacteria. So these researchers developed a question: which is better, fertilizer or crop rotation? What they did is they ended up with 4 different groups. They had their control group, which is going to have the same kind of plants in it. It's again going to be watered the same amount. It's going to get the same amount of sunlight. All of that stuff's going to be held constant, but it doesn't get crop rotation and it doesn't get fertilizer. Then they're going to have a test pot that gets a half dose of fertilizer, a test pot that gets a full dose of fertilizer, and then test pots that are getting crop rotated with legumes to find out whether or not the legumes do better than the fertilizer, which they're thinking is going to do better than the control group off over here. Okay, the data that they collected, we're just going to look at year 1 at first. So this is the control group's growth, half dose of fertilizer group, full dose of fertilizer group, crop rotation group. What they found after 1 year was that their hypothesis was rejected. They thought that crop rotation was going to provide them with the best yield, and it didn't. The full dose of fertilizer is what actually gave them the best yield of their winter wheat and their pigeon pea that they were rotating with. However, science doesn't usually stop with just one experiment, so then they came up with a new question, which was, if you sustain crop rotation, will you eventually get better yield than you do with fertilizer? So this is the same setup after year 2. Notice that crop rotation is now doing better than fertilizers and the control group. This is year 3. It did way better than our fertilizer groups and our control group up over here. And so after a sustained experiment over several years, they found there is definitely a benefit to crop rotation. This kind of experiment has a control group. They did replicate their results. In other words, they never just tested one pot. They would have, like, I'm just going to make up a number. They would have 5 of these pots, 5 of these, 5 of these, because sometimes things just happen in one pot, and so you always want to replicate your results too. Here they showed you 2 pots, but they would have the same number of those that they had of the other ones. One of the other ways that this can be used is, let's say you've got a farmer and they're considering crop rotation. Let's say they had 100 acres. Would you want them to start rotating their crops on all 100 acres at one time? No, because that first year they'd lose money and then they would not be behind crop rotation anymore. Instead, you have them just do a little bit at a time, because eventually it will pay off in a really big way. It just doesn't at first. And so this data can be used in multiple ways. Yes, crop rotation is better, but it's only better in the long haul. Already did that. More things, stuff and things. Okay, so I mentioned replication just a second ago, and I want to come back and talk about the importance of them. Back in the 1990s, this guy, his name is Andrew Wakefield, he wrote up a letter to the editor in a journal called The Lancet and said that he was finding that the MMR vaccine was leading to an increase in what he called abdominal autism. Now, for any of you that know what autism is, it's a nervous system spectrum disorder that is characterized by social abnormalities. There can also be visual and auditory issues that go along with this, and there can be a nonverbal component to it. When I say spectrum, I mean there are some people who are very high functioning, and you might not even notice the average person that is high functioning who has autism, but there's also people on that spectrum who will never be able to live a life on their own. He called it abdominal autism, which was basically stomach cramps, nausea, and diarrhea. It is not the same thing that you guys think of as being autistic, although even people with the neurological issues that go along with autism, they can have GI symptoms as well. He did not replicate his results, and when other scientists tried to replicate his results, they were unable to come to the same conclusions that he did. So by submitting to scrutiny, they would have found that his experimental setup was completely flawed, but unfortunately he got published before that scrutiny actually happened, and unfortunately even to this day, nearly 30 years later, people still believe that vaccines cause autism, and that has led to deaths, literal deaths, from people who didn't get vaccinated and then ended up getting diseases that can kill you and were completely preventable. So it's very important that replication happens, and it should happen before publication even occurs. Unfortunately, that's just not the way the publication industry is working these days. Okay. One of the other things that you should be able to do is you should be able to graph your data. So what I have us, we're going to set up here is something that we're actually going to be, well, sort of kind of doing in lab later on, since we're not really on campus and in person. We're going to test the fermentation rate of yeast in devices that are called respirometers. What we do is we take the yeast, we mix them with a fuel, and then we look to see how much carbon dioxide gets produced. We would do that at multiple different temperatures to find out whether or not temperature influences the rate the yeast can actually, ferment at. So here's the data that we could collect for this. What I want you to notice is that there's 3 different temperatures that we put the yeast at so that they could ferment. What you would need to do if you wanted to graph that data is take all of the replicates at one temperature and then average that data together. And so grab your phone, it has a calculator on it, and take the average of those numbers, and you would find that these guys on average produce 2.3 millimeters of carbon dioxide. Then take the next 3 and average those together, and you would find that they produced 4.3 millimeters of carbon dioxide. Take the last 3. They produced 8.3 millimeters of carbon dioxide. Once you have the average, at that point you can graph your data. When you graph your data, trying to figure out if I can do it on this screen or if I need to do it someplace else. I'm going to do it over here. Okay, so this is the x-axis, this is the y-axis. On the x-axis, you put your independent variable, which again is the thing that you have control of. What we had control of here was the temperature. We had 3 different temperatures. So we've got 20 degrees, go away, 25 degrees, and then 35 degrees. So you would go ahead and put those on there. Why are you hassling me, dude? 20 degrees, 25 degrees. I really have no idea why that button keeps coming up, but it's bugging me. And then 35 degrees. And that's in degrees Celsius. Okay. The y-axis gets your, I really have no idea why it's hassling me. Gets the dependent variable. Why are you picking on me, dude? I clicked what I'm supposed to click. It's just really not enjoying me today, I guess. I'll try it with the mouse, although it'll probably be even messier. Okay, so this is going to be our dependent variable. The dependent variable is the thing you measure, which in this case is the amount of carbon dioxide that gets produced. So let's say, I want it to go up to 8. So this is going to be 2 millimeters, 4, 6, 8, and you would label that and do it neater than I did. Okay, then 20 degrees Celsius on average did 2.3, and so you would go ahead and create a little bar graph. Pew. All right, and then 4.3 for the next temperature, so that's going to be right about here. And then last was 8.3. By graphing the data, you're able to see visually much faster that, yeah, temperature totally has an impact on how fast yeast can ferment. The warmer it is, the faster the metabolism is, which means the faster they can ferment the sugars that are present. We'll practice graphing this data in the lab on the scientific method too, so don't think this is the only time you're going to see that stuff. From here, so this completes the introduction to biology notes. What I'm going to do is post a quiz in Blackboard so that you can, well, Blackboard for my Tarrant County students, Schoology for my Hill College students, but you guys can take that quiz. Remember, you can use your notes to help you out with the quiz, but you should be good to go for the introduction to biology chapter.